Have you ever wondered how water is transported to the flowers of a plant against gravity? Plants use capillary action to move up water from the soil through the rest of the plant. Capillary action is the movement of liquid along a surface of a solid caused by the attraction of molecules of the liquid to the molecules of the solid. Water molecules are attracted to the molecules of inside of the stem. It is this exact attraction which helps force the water up from the ground and around the plant. We come across three forces that are involved in the process of capillary action, namely adhesion, which allows the water to stick to the organic tissues of the plant. Second is surface tension. This helps to hold the water molecules together as they are being transported throughout the plant, and cohesion, which keeps the water molecules together. For capillary action to work, the adhesion force between the water and plant tissues must be stronger than the cohesion between water molecules. To understand this better, let us conduct an experiment. Get into your science mode and let's dive in. For today's demonstration, we would be requiring water, food colors, tissue, a pair of scissors, spoon, 4 glasses, and white flowers, preferably roses. Let's start with cleaning our flower stems. Try to remove the excess leaves as well as the old wilted petals and keep all the roses ready for the experiment. Be mindful when you cut the stems of the flowers. Try to cut the stem at an angle. Cutting the stems at this angle will allow for better water intake as they won't be sitting flat on the bottom of the glass. Now that our flowers are ready, let us prepare the colored water solution. In each of the glasses, add some water and mix the food colors of your choice. Here I have used pink, blue, orange and mixed pink and blue, making a purple shade. See to it that the color completely dissolves into the water and no lumps are found. Place the flowers carefully into the colored water glasses. Now, place the setup in a cool, lit and ventilated room and observe the change in the colors of the flowers. Now that it has been nearly 4 hours, we can observe that there has been significant change in the color of our flowers as well as the stem. Let's cut into the stem and observe the changes accordingly. We always wonder how plants drink water. The water travels up the stem and into the petals of the flower. Now here in our experiment, our flowers did not have roots anymore, but they were still able to pull water up from the cup into the stem and up through the flower to its petals. It is only through capillary action that plants are able to defy gravity and pull water up and into their flowers. We can also observe clearly the branched organization of the non-well-developed vascular bundles which are present in the petals of a flower. The water which has been colored has clearly traveled all the way from the stem to the petals. Through this experiment, we have concluded about how water moves up with the help of tubes present inside the stem called as a xylem. I hope this video was helpful in making you understand about the capillary action in flowers. Also, if you ever need any color in your flowers, you know how to get one. Thank you so much. Do you think it is possible to make water run uphill without using electricity? Born in 3rd century BC, Archimedes was a Greek mathematician who designed what we now call the Archimedes screw. It was used to pull up water from a low-lying water body. It is one of the earliest hydraulic machines. The design is so effective that it is still being used today. The Archimedes screw is a form of positive displacement pump which traps fluid from a source and then forces the fluid to move to a discharge location. To understand this concept better, let us conduct an experiment. The materials that we will be needing for today's experiment are one PVC pipe which is at least an inch in diameter and at least a foot long, clear vinyl tubing which is at least 1/4 inch in diameter and at least 2 feet long, duct tape, a pair of scissors, 2 containers for water, an empty bowl to elevate one of the containers, food coloring. The bowl that shall act as the low-lying water body adds some food coloring to the water. This shall help us to study the movement of the water clearly. Now we shall construct our own Archimedes screw. Attach one end of the vinyl tubing to one end of the PVC pipe with duct tape. Ensure that the opening to the tubing is open and not blocked by the tape. Tightly wrap the tubing around the pipe in a spiral manner. Attach the tubing to the other end of the pipe with duct tape and again be careful to not block the opening of the tubing. Try to evenly space out the tubing along the length of the pipe and secure its position. Also make sure that the vinyl tubing is not being squeezed or pressed too hard when you are wrapping it around the PVC pipe, as this might affect the flow of water. If required, try using some extra pieces of duct tape to secure the vinyl tubing around the PVC pipe. Now we start with our experiment. Firstly, elevate the second, which is the empty container, at a higher position than the first container. Place one end of your Archimedes screw in the lower container of water and align the other end over the upper container. Rotate the screw so the bottom end of the tubing scoops water with each rotation. It should go underwater and then come back above the surface with each revolution and not remain completely submerged the entire time. You should see your tubing start to fill with water after a few rotations, and as the water keeps climbing up, it eventually falls into your empty bowl. So what do you think made it possible for the water to climb from the lower bowl to the upper bowl? Yes, it was possible using the Archimedes screw. Let me explain this concept now. The Archimedes screw is made up of a hollow cylinder and a spiral part placed inside. For today's experiment, we had put the spiral part outside the cylinder. One end is placed in a low-lying fluid source and the other end is tilted up into a higher discharge area. To move water, we need to rotate the screw. As the screw moves, it scoops up a small amount of water into the first pocket. On the next turn of the screw, the first pocket of water moves to the second pocket and a new scoop of water enters the first pocket. This motion continues and eventually the first scoop of water comes out at the other end. That you were able understand the concept, the working model, the benefits and the uses of the Archimedes screw. Thank you for watching. Have you ever wondered how all the food that we eat gets digested in our body? There are little molecules that do a lot of work that are called as enzymes. Enzymes are proteins that act as biocatalysts that help speed up chemical reactions like digestion and metabolism. They build some substances and break others down. All living things have enzymes. There are about 1,000 different enzymes in our body that work to keep us healthy and active. Enzymes are very specific and only react with chemicals called as a substrate. Today we shall be studying about an enzyme called as catalase. Catalase is a common enzyme found in nearly all living organisms exposed to oxygen. It is a crucial antioxidant enzyme in living cells which plays an important role by breaking down hydrogen peroxide and maintaining the cellular redox homeostasis by protecting them from oxidative damage. Catalase helps in preventing the damage by breaking up hydrogen peroxide into harmless water and oxygen. This enzyme is present in commercial yeast. Today's experiment shall explain how this enzyme activity takes place and what makes the activity possible. The materials that we will be requiring for today's experiment are liquid dish soap, a tablespoon of dry yeast, hydrogen peroxide at 6% concentration, glasses, teaspoon, tablespoon, warm water around half cup, label makers, pen, tissue roll and some food coloring. We begin our experiment by preparing our yeast solution. Take a bowl of warm water and dissolve the dry yeast. Let the dissolved yeast rest for at least 5 minutes. Label the glasses from 1 to 3 and mention the amount of hydrogen peroxide that has been added. We add no amount of hydrogen peroxide to the first glass. We add half tablespoon and 2 tablespoons of hydrogen peroxide to the second and third glass respectively. This shall help us study the enzyme activity well. To all the labeled glasses, add 1 teaspoon of liquid dish soap. To each of the labeled glasses, add a drop of food color and stir well. This just makes the experiment more fun. After adding the soap and the color, mix this solution very well. As we will be handling hydrogen peroxide at 6% concentration now, be careful while using it. Any spill on your skin should be rinsed with plenty of water. As mentioned earlier, as per the label makers, we shall be adding the same amount of hydrogen peroxide to each of the glasses respectively. Nothing shall be added to glass 1. After adding the hydrogen peroxide to each of the glasses, stir well. As we come to the main segment of our experiment now, please place tissues under all of your glasses as this could get little messy and this will also make cleaning the entire setup easier. Add 1 tablespoon of the previously prepared yeast solution to each of the glasses and just observe the reactions. Are you wondering how this happened? The answer is, when the enzyme catalase comes in contact with its substrate, hydrogen peroxide, it starts breaking it down into water and oxygen. Oxygen is a gas and will always try to escape the liquid. The dish soap here that we added was able to trap the gas bubbles, which results in the formation of this stable foam. As long as there is enzyme and hydrogen peroxide present in the solution, the reaction continues and the foam is produced. If there is no hydrogen peroxide present, the catalase cannot function, which is why glass 1 had no bubble or foam production. The presence of hydrogen peroxide makes the catalase to be active as observed in the other glasses. In fact, the catalase reaction is dependent on the substrate concentration. If you have an excess of enzyme but not enough substrate, the reaction will be limited by the substrate availability. As a result, glass 3 had more substrate, so we observed more foam production. A catalyst does not take part in the reaction itself. So how does it really work? Each chemical reaction needs a minimum amount of energy to make it happen. This energy is called as the activation energy. The lower the activation energy, the faster it takes place. Enzymes have the ability to lower the activation energy of a chemical reaction. The compounds or molecules the enzyme reacts with are called as substrate. The enzyme pocket has a special shape so that only one specific substrate is able to bind to it. Once the molecule is bound to the enzyme, the chemical reaction takes place. These enzymes are very specific. On a cellular level, if there is imbalance of antioxidants, then the physiological condition is called as oxidative stress. A catalase is one of the crucial antioxidant enzymes that mitigates oxidative stress to a considerable extent by destroying cellular hydrogen peroxide to produce water and oxygen. The deficiency or malfunction of catalase can result in many illnesses such as diabetes, hypertension, anemia, vitiligo, Alzheimer's, Parkinson's, bipolar, schizophrenia, and cancer. I hope this video was able to help you understand about enzyme activity, the enzyme catalase, and its importance. Thank you for watching.